Brockport. He's going to talk to us about using fatty acid signatures to discuss Lake Trout at Cayuga Lake.
Thanks, Andy. Hey, everybody, thanks for coming to my talk. I'm a graduate student at Brockport. I'm studying under Jacques Rinchard in his fatty acid lab, and I did a project on lake trout feeding habits in Cayuga Lake using fatty acid signatures. So to start out, I'll introduce my lake that I studied. Cayuga Lake, it's the second deepest and longest of the Finger Lakes, located just north of the city of Ithaca. Not much is known about the lake trout feeding habits in the lake. Stuff's been done as far as diet analysis in stomachs, but nothing has been done as far as fatty acids, so I'm kind of pioneering the field on Cayuga Lake with the fatty acid work. We've determined through historical data that the alewife and round goby and rainbow smelt. Round goby is a relatively new invader to the lake. I first caught this round goby here in 2013, June 26th to be exact, at Cayuga Lake State Park. So that really sparked my interest into checking out what's going on, and things just kind of worked out the way they did as far as getting into the field. So fatty acids, basic background fatty acids, they're long chains of carbon molecules with a carboxyl group affixed to the end. And I looked at a total of 27 of these to make up total fatty acid signatures, in which we use to assess the diets of each fish that I studied. Fatty acids are important in food web ecology because they're conserved across feeding meals. So technically you are what you eat. They're also important in membrane fluidity of cells, and they're a high energy supplement in diets of organisms. So predator-prey relationships are pretty well studied using fatty acids in marine systems, and over the past 5-10 years have been really well implemented in the Great Lakes. Last year, a study was done doing similar stuff to what I'm doing in Lake Ontario, and I hope to find similar results in Cayuga Lake, and I will discuss that in the next coming slides. So my objectives were to look at the two prey items that I collected, round goby and alewife, and to assess whether or not their fatty acid signatures were different, in order to then compare them to lake trout. Because if they aren't different, unique between the species, then if you try to compare them to lake trout, then it's just going to look like they're eating everything, and you can't really decipher any differences. So then, assuming I collected different fatty acid signatures in prey species, I then compared them to lake trout. My methods were to collect prey fish from around Cayuga Lake, 4 sites, Cayuga Lake State Park, which is on the southwest shore, Deans Cove State Boat Launch, Long Point State Park, and Myers Park, which is a state, or I think it's town of Lansing owns the park. And then all my lake trout were collected from Cayuga Falls State Park, thanks to DEC Lake Erratic Tank. So my capture methods, gill netting, of course, for lake trout at Cayuga, and I used inshore seining, using a 25-meter seine with a big old cod end in the middle of it. And it was relatively successful. This was a haul we took from Deans Cove, right along the break wall, outside of the boat launch area, the marina area. And there's just all kinds of stuff we were picking up. We caught brown trout, we caught alewife, we caught gobies, we caught bullhead, everything. Pretty much everything you could think of that would be there, we caught it that day anyway. So for my chemical analysis, I used a method of lipid extraction used by Fulch et al. I won't get into details with it, it's kind of gross and nasty, but after I did that, I used the fatty acid transmethylation method to then make the fatty acids readable through our gas chromatograph and mass spectrometer. So by running these transmethylated samples through the GC and getting an output using the mass spec, we got these chromatograms, which each peak will represent an individual fatty acid in your individual fish. So I did almost 800 of these for all the fish I collected, and each individual chromatogram represents one fish. So my statistical analysis that I ran were a non-metric multidimensional scaling, NMDS for short, to compare temporal and spatial differences between or within species of crayfish. And then I compared the actual crayfish differences using the same NMDS or method, and then also compared the prey to the predator using the NMDS method. An analysis of similarities I used to assess whether or not there were any significances between the individuals I was comparing, and the similarity of percent analysis I used to assess which fatty acids were actually causing the differences between fish. So this is a total of all the fish I collected and used in these results. Of course, 4 sites, 3 sampling seasons in 2 years, a total of 714 crayfish in total, and 60 lake trout, 30 from each year. So as I said before, the first thing I was going to do was compare the 2 cray species and see whether or not there was a significant difference between fatty acid signatures. And according to my NMDS and my NSM, there was a significant difference. The R value is represented on a scale of 0 to 1, 1 being absolute difference and 0 being no difference at all. They're exactly the same. And with an R value of 0.596, they're significantly different, and you can see in the NMDS that spatially they are grouping in 2 different areas of the NMDS, and that signifies a significant difference. My SIPR results, I have 4 fatty acids I looked at. 18, 1, and 9 had highest concentrations in alewife, and the other 3 were all higher in round goby than in alewife. So those 4 fatty acids are really making up the majority of what's driving the difference between the 2 species. This is the same NMDS plot, but instead of species comparison, I added in the factor of the season, and I got a significant difference in the seasonal variation within the round goby, but that significant difference is less important than the fact that the 2 species are different from each other. So it's an interesting result that I got, but there's not a lot of weight in it as far as driving the 2 differences between the species. So I just thought I'd share that and explain that it was an interesting result, and even though there's a difference within the round goby, it doesn't matter really as far as moving forward and comparing them to the lake trout. So I also did the same thing with the lake trout, comparing the 2 years of collection, and there was also a significant difference here. 2014 in green and 2015 in blue. There's some separation, but again, that difference, it doesn't outweigh the fact that they're very similar, having about a 94% similarity within that group of fish. There's 3 fatty acids that drove the difference. The main one being carbon 16 was higher in the 2014 fish, and 18, 1, and 9 and 16, 1, and 7 were higher in the 2015 fish. I'm not really sure why they're different. I can only speculate, but it's a result that I got, and I'm going to discuss it more in my thesis as I continue to write. So, as I said, I did find a significant difference between my crayfish, and I went ahead and compared them to a lake trout using my 3 statistical analyses. And I found that, on average, lake trout are much, much more similar to alewife than round goby. as I said before, my R values are represented by a scale of 1 to 0. 0 meaning there's absolute similarity, 1 meaning there's absolute dissimilarity, or the 1 meaning there's absolute similarity, right? So absolute dissimilarity. Round gobies had R value of 0.77, meaning that they're more different from lake trout than the alewife, which had an R value of 0.52. And as you can see in my NMDS plots, spatially, lake trout grew right at the end of what are mostly the fall alewife, which is the same season in which I collected the lake trout. So, in conclusion, there are differences between the two crayfish I collected in fatty acid signatures, driven mostly by 18.1 and 9, which is higher in alewife, and 22.6 and 3, which is higher in round goby, with a significant R showing the difference. And any intra-species variation within my species is outweighed by that variation between my species. And then, of course, lake trout comparison. I found that they're different from both species as far as the R value goes, but when comparing the gobies to the lake trout and the alewife to the lake trout individually, I found that the round goby is less similar to the lake trout than the alewife. So I guess in speculation with that, I would think that lake trout are probably eating gobies, but by and large, the majority of what they're eating is likely alewife and maybe other species. The reason I didn't include the smelt in the study, as I discussed they're a major prey item in Cayuga Lake in the beginning of my presentation, was because I couldn't really catch it. In speculation for that, I think the fishery of smelt in Cayuga Lake is really, really small. Just couldn't get it. I caught maybe 5. So with that said, I'd like to thank everybody who helped me with my project, my funding sources, and of course the AFS committee for having me here today to speak. And I'd like to leave you with this really bad Photoshop and any questions you might have.
Sometimes we have concerns when we're stocking, say, brown trout or something into one of the finger lakes. We have some evidence that suggests maybe the large lake trout populations are gobbling them all up before they can recruit to the fishery. Do you think these types of methods are something that could
Absolutely. Absolutely. If we took the fingerling browns and some of the wild lake trout, maybe pre and post release, we could look and see if they're actually eating them. We could compare the diets. I guess it really depends on how much alewife is in the system, because they're hammering alewife. Yep.
So Jeremy, when you sample the lake trout and you get your fatty acid results, what time period does that cover for what it was eating?
I've been told it's about a 3-month period to incorporate fatty acids of what they continually eat into their signature. It may be a little less or a little more depending on how often they eat and other factors like water temperature and metabolism and things like that.
But it's a long-term integration.
Yes. Whereas something like stomach analysis, immediate, it's maybe that day or that week, and as soon as it's digested, you have no idea what they ate. So it makes it a valuable tool to management, because if you're trying to figure out what they're eating, you'll know in a snapshot if you're looking at stomachs, but if you look at this, you might know over the course of months.
I think that's interesting. It's a 3-month transition as it goes into the tissue and out of the tissue. That doesn't necessarily jive exactly with the seasonality you saw in I believe it was around Gobi, right? That you were actually able to see separation
Right.
within a season.
Well, no, that was within 3 seasons. I'll go back.
Right. But there were differences between spring, summer, and fall. You can almost see 3 signatures there.
Right. So I guess you have to look at the definition of the season. For me, I'm thinking like every 3 months, right? 4 seasons is a year, 3 months.
But that's also assuming that they turn on and off a switch of prey or eat something different.
You really have, I couldn't say for sure. You'd have to look into the ecology of inverts, because I'm assuming they're eating mostly inverts, maybe barbel fish. The gobies are so voracious, they eat basically anything they can put in their mouths. And if it's there in front of them, they're probably going to eat it. It's something I'm going to speculate on as I go forward, but yeah, that's a really, I'm glad that you raised that to a point.
I got a question.
Sure.
Do you have any access to seasonal diet data that the DEC or through their surveys? Anybody have that kind of data that you could perhaps match up?
I don't, and unfortunately, it's really kind of challenging to assess anything that's not a fish as far as fatty acid work goes, because you'd have to, it's by the individual, so if you take like a scud or something and grind it up, you wouldn't get a good concentration of the fatty acids by doing the method that we do. It would have to be really small scale, and what we do is evaporate down the raw fat using chloroform and other mixture solvents, and you get a really almost an infinitesimal tiny little bit, and being able to get a significant reading on it is challenging. Even with fish that are 10 millimeters, sometimes you don't get enough lipids out of the sample to be able to run it.
Is there any advantage, or have you given any thought to using sort of as an additional line of evidence looking at carbon stable isotopes?
It's something I'm familiar with, but we don't have access to that in the lab that we work in. We'd have to outsource it, and unfortunately, I didn't add it in to what I was working on. But it would be useful to definitely compare the carbon and stable isotopes to what I did here.